Hello, hello everyone. My name is Dr. Swapnil Desai, and today's date is 20 December 2025. I will be presenting chapter 33, Heme Synthesis and Degradation from Harper Illustrated Biochemistry. Uh, Heme is an iron-containing porphyrin compound that plays a crucial role in human physiology. It is an essential component of hemoglobin, myoglobin, cytochrome, catalase, and peroxidase. Without heme, oxygen transport and cellular respiration cannot occur efficiently. Heme synthesis mainly occurs in two organs, in liver and bone marrow. In liver, where heme is used for cytochrome enzyme, and in bone marrow, where it is required for hemoglobin synthesis and red blood cells. Uh, Now, overview of heme biosynthesis. Heme synthesis is a multi-step process that taken place into two cellular components, mitochondria and cytosol. The two major precursors required are glycine and succinyl-CoA enzyme, and the pathway requires vitamin B6, also known as pyridoxal phosphate, as a cofactor. And rate limiting enzyme, there is a ALS synthesis is a rate limiting enzyme for this. In this first step and most important step of the heme synthesis occur in mitochondria, and in this glycine combines with succinyl coenzyme to form delta amino levulinic acid or ALA, and this reaction is catalyzed by the rate limiting enzyme ALS synthesis. Heme itself regulate this step by feedback inhibitions and preventing excessive heme production. Cytosolic step. ALA is transported from the mitochondria into cytosol. Here, two molecules of ALA condense to form porphobilinogen and catalyzed by ALA dehydrase. Porphobilinogen is converted into hydroxymethyl labine, and which is cyclized to form uroporphyrinogen 3 and later corpopyrinogen 3. In final mitochondrial step, corpopyrinogen 3 re-enters in the mitochondria, where it is converted into protophyrin 9, and the final step involves insertion of ferrous iron into protophyrin 9. This reaction is catalyzed by peroxidase and producing heme. Regulation of heme synthesis. Regulation occurs mainly in the level of ALS synthesis. In the liver, certain drugs, alcohol, and fasting can increase enzyme activity, which explain drug-induced porphyrins. In erythroid cells, regulation is influenced by erythropoietin and iron instability. Iron availability. Porphyrias. Porphyrias as a group of inherited or acquired disorder caused by defect in enzyme of the heme synthesis pathway, and they are classified into acute intermittent porphyria and cutaneous porphyria. Acute intermittent porphyria which affect the nervous system, and cutaneous porphyria which cause photosensitivity. Acute intermittent porphyria is caused by deficiency of porphobilinogen deaminase. This leads to accumulation of ALA and porphobilinogen. Patient typically present with severe abdominal pain, peripheral neuropathy, and psychiatric symptom. Importantly, photosensitivity is absent in acute intermittent porphyria. Now, heme degradation. All red blood cells are destroyed by macrophages in the spleen, liver, and bone marrow. Heme is converted into biliverdin by the enzyme heme oxidase and releasing iron and carbon monoxide. Biliverdin is reduced to bilirubin by biliverdin reductase. And then bilirubin form in macrophages is unconjugated and lipid soluble and toxic. It is transported into bloodstream bound to albumin and delivered the liver. Now, bilirubin conjugation. In hepatocytes, bilirubin is conjugated with glucuronic acid by UDP glucuronyl transferase, and conjugated bilirubin is water soluble and excreted into bile. In the intestine, conjugated bilirubin is converted by bacteria into urobilinogen, and some is excreted as a stercobilin in feces, and giving stool its brown color. A small amount of reabsorbed and excreted in urine as urobilin. And last, jaundice. Jaundice is yellow discoloration of skin and sclera due to elevated bilirubin levels. There is 3 types: prehepatic jaundice, hepatic jaundice, and post-hepatic jaundice. Each type has characteristic laboratory findings. Important clinical conditions include neonatal jaundice, Gibbler syndrome, Kigler-Najjar syndrome, Dubin-Johnson syndrome. Understanding bilirubin metabolism help in diagnose this disorder, and this is the clinical correlation with this. And in the last conclusion, heme synthesis and degradations are essential biochemical process, and they involve precise regulation, multiple enzymes, and distinct cellular compartment. Disruption of this pathway lead to clinically significant disorders such as porphyrias and jaundice. Thank you.